All right, people, let's knock out amino acids. Let's just be done. Okay, something I forgot to mention up here. So if you do not like water, if you in fact hate water, because the other things are bonding mostly by hydrogen bonds, but let's say you hate water. So you have to interact by, let me just put it here, hydrophobic interactions. So you hate water, you're going to have to interact with yourself. That's basically what's happening. You don't have any hydrogen bonds because you hate water, then you're going to have to interact with some hydrophobic, some other people that hate water too. You're going to have to do your own thing. Okay, so secondary protein. We're done with the beads on a string business. Now we're going to talk about how this thing is going to coil around and do some other stuff. So the alpha helix, it's going to look kind of like a DNA helix molecule, like a telephone cord, like a slinky. Pick something that's like that. How is this happening? How is this thing bonding? It is bonding. Guess. Just guess. Come on. Come on. You can guess. I'll give you a hint. Hydrogen bonds. Yep. I told you. If you needed to guess hydrogen bonds. So that's basically what's making this helix. So this helix molecule is brought to you by hydrogen bonds. The hydrogen bonds stabilize interactions. So the other one is the beta pleated sheet. Say it 10 times fast, just do it because it's fun. The hydrogen bonds stabilize this business here. This is a beta pleated sheet. It's like an accordion, something like that. These hydrogen bonds are stabilized between protein chains. Oh, where am I writing that? Why is it not happening? Okay, between protein chains. So this is what's happening. You have hydrogen bonds. Of course you have hydrogen bonds. And they're sort of interacting between the protein chains. So you have some up, some down, like a little accordion, and they look all pretty. And again, hydrogen bonds are involved. The protein chains lie side by side, so that you can alternate them. So that the altering chains run like up and down like this, kind of. I don't know. Does that make sense? From the one terminal to the C terminal to the one terminal to the C terminal. And this is known as anti-parallel. Again, I am not even trying to spell that correctly, an anti-parallel arrangement. A stack of these pleated sheets illustrate how the R groups point to the above. Really, it's like I'm not even trying to spell, and below the sheets. So you have a pleated sheet illustration, and you have one of them going above and then below, and then above and then below in this sort of anti-parallel arrangement. And so they're sticking up above and below, and it's sort of creating a folding that folds it together like an accordion. It's called a beta pleated sheet. That is secondary structure. So primary structure is this, and then is it going to kind of do like that maybe? Or is it going to do like this, like a slinky? So that's secondary structure. This is how it's going to kind of coil up and be really small. Now, how is that thing going to interact? I've gone too far. How is that thing going to interact? This doesn't. So now we're into tertiary proteins. Now it's going to just really tighten up. It's already like a coil, and now you're going to tighten it up even more. This is called tertiary structure. Tertiary structure. I think it's just tertiary protein structure is what I want there. So this is called tertiary structure. Basically what's happening is we're folding it over on itself. Now we're going to use some disulfide bridges. We're going to use all the interactions we can to just scrunch that thing up, make it much smaller. Each protein folds in a distinctive manner that is basically determined by, I have no idea why that's not spelled right. So the manner that it folds up in its secondary, but primarily it's tertiary, that doesn't make sense. The tertiary structure is determined mostly by its primary structure. So mostly by the beads on the string. Remember I said you screw one of these up, when one of these gets mutated somehow, it's going to mess up the whole thing. So it's not going to, maybe it'll still do this, maybe, but it won't bind properly and fold over on itself. It won't make the proper shape. Remember the Pac-Man, if his mouth isn't exactly right, the enzyme's not going to work. If you're folding up into a hemoglobin protein, it's not going to work. You're going to have sickle cell. It's going to be bad. So the primary structure is super important to the tertiary structure. The tertiary structure is drawn in a style that shows the combination of alpha and beta regions. So you're going to have both regions working together. And this is mostly the superfund disulfide bonds. The disulfide bonds are really important here. They're going to move things together. So here is an awesome diagram of what it all looks like. So here's your primary, is just going to be the little ones inside. Then you're going to have a helix. Then you're going to have all these bonds and bridges. And here's your disulfide bridge, which is going to be down here. So kind of stare at that for a while. So that is your secondary structure, quaternary structure. So this is, now you've got your beads on a string. You fold it into a helix. You've sort of refolded it over on itself. And now you're bonding it with somebody else. So this is primary, secondary, and tertiary, right? You have a primary structure, which is the beads on a string. The secondary structure, it's in a helix. The tertiary structure, it's like all these like disulfide bridges and all this awesomeness is happening. And you got this like wad of awesome protein. Now you're going to bring in another protein and you're going to bond it together. That is quaternary protein structure. Now, what determines this? The way in which the entire chain is coiled and folded into this is dependent on not actually primary, but tertiary. Tertiary is protein structure, is basically going to determine your second thing there. These polypeptides are held together. Polypeptides are primarily held together by what forces? I'm sorry. This should be quaternary. I'm sorry. We have moved on to quaternary structure. Quaternary structure. Quaternary protein is the way in which so now we're on to quaternary 4, the fourth stage. Polypeptides are held together by non-covalent forces. So these are non-covalent forces. You want to be able to pull these things apart if you need to. So, are primarily held together by non-covalent forces. However, covalent forces and non-amino acid portions are also incorporated into the whole thing because this is a big deal. You're putting two things together. So the quaternary structure of a hemoglobin. So hemoglobin consists of 2 globulin chains. Boom, 2 globulin chains. Very complex how they're like all put together. Bonded primarily by hydrophobic interactions. They're water haters. They don't like water. And so a lot of their hydrophobic interactions are happening. So I hope that gives you primary, secondary, tertiary, quaternary, because that's what's going on. Now we are just going to review it a bit. So primary structure, give it to me, guess. Is the sequence connected? How are these things connected? Come on, you know. Peptide bonds with the space in between it. Peptide bonds, that's how everybody's coming together. Peptide bonds. The bond that doesn't break is the peptide bond, people. Okay, secondary structure. So primary structure, peptide bonds. Secondary structure is going to be made up primarily of a bunch of loops and coils that connect the segments. So is it going to be a loop? Is it going to be a coil? Is it going to be accordion? Is it going to be a helix? What is happening? This is held together by hydrogen bonds, primarily hydrogen bonds. Let's make that red, circle it, underline it. The hydrogen bonds. And the hydrogen bonds are happening between the carbonyl group and the amino acid group. Yeah. That is what is happening. So that's primary, secondary, tertiary, review. Tertiary is the what? Tertiary is the folding. Now we're like all folding it together. Folding. This is folding everybody all together. It's the folding of a protein into a specific 3-dimensional shape. So now it looks like a big giant glob, but it's actually specific. This is happening typically, what is this? Non-covalent interactions. So here we have peptide bonds, hydrogen bonds, non-covalent interactions, all kinds of stuff happening here. Okay, quaternary structure. Also what's happening is, disulfide bonds. This is also what's happening. Don't forget the disulfide bonds, they're important. Quaternary structure is 2 or more. So now 2 or more friends. So now we've invited some other proteins to the party and we are making a giant, huge protein. We've invited some more polypeptides to the party and we're making a big, giant thing. Okay, let's talk about one more thing. Chemical properties of proteins. Can we do it in 5 minutes? I think we can. Okay. Digestion. Let's talk about digestion. Digestion hydrolyzes. Hydrolysis. Digestion hydrolysis hydrolyzes peptide bonds, yielding smaller. So it breaks it up. So we put them together. We're just going to break that. We're just going to break it up. That is digestion. Denaturization. We talked about how to denature things. Denaturization. Denature. Let's just put denature. Denaturing. Denature is the loss of secondary, tertiary, and quaternary proteins. When you denature a protein, you are not breaking this up. This bead, these beads on the string, those guys stay together. You are not breaking those things up. If you want to break those things up, you have to use hydrolysis. You have to pull those peptide bonds apart. The only thing that can do that is a hydrolysis reaction. If you want to just unfold it, that's fine. That's secondary, tertiary, and quaternary structure. You can do that in a number of ways. You can denature the protein. But if you want to actually pull these things apart, you have to use a hydrolysis reaction. It's much more serious. Okay, let's rock. Okay, solubility is often decreased in denaturization. You can look at this. We can look at it in lab. Solubility is often decreased. So enzymes lose their catalyst ability. This is mostly how things get denatured. They're not able to carry out biological functions because they are denatured. Most denaturization is irreversible. Once you denature something, it is denatured. You cannot get your egg back into the egg shell. It's denatured. Renaturization is sometimes possible, but it's usually not going to happen. All information to determine protein shape is present in the what? Give it to me. Come on. Primary structure. So you're not messing with the primary structure unless you're hydrolyzing the reaction. You are not messing with it. You might be messing with this folding and how it's folded. It's going to unfold and get all weird and denatured, but you still know what was there. You still know the primary function, and that is really important. Okay, how do you denature things? I'd like to denature some proteins today. What should I do? The first way is to heat it. Just think about, like if I ask you on this test, how can you denature an egg? Let me tell you a whole bunch of ways. The first way, you can heat it, right? What does that actually do when you heat up the egg? The weak side chains. It's heating, it's shaking it. The side chains attractant are disrupted. So you're shaking everything, you're pulling those side chains apart. They're like, what? Get away. The second way, mechanical agitation. You can froth it. If you've ever like whipped egg whites and made like a froth, you're denaturing it. You're denaturing your egg whites. Denaturization of proteins at the surface of the air bubbles stiffens. So it stiffens up your egg. If your mom has ever made you do this by hand, you whip and whip and whip and whip and whip, and finally it becomes stiff. You're denaturing the egg whites. That is another way to do it. You just beat it. You just beat it until it's stiff. And then once it's stiff, it can't go back. Detergents, you can soap it. Even a low concentration of detergents can disrupt the association. What are we disrupting? What's detergent disrupting? Hydrophobic or hydrophilic? Hydrophobic. So those hydrophobic side chains, they're getting disrupted by detergent because detergent's making everything weird. And it's getting rid of, that's why we use it on proteins, right? That's why it gets grease out. That's why we use Dawn on your greasy pan. That's another way you can think of it. Okay, how are you going to get the grease out of the pan? How are you going to disrupt the stuff? You're going to heat it. You're going to scrub it. You're going to use detergents. Okay, organic compounds, another super fun way. So another way you can do it is you can actually compete for the binding sites. So these polar solvents interfere with the hydrogen bonding. It's all about hydrogen bonding, and they're actually competing for those bonding sites. So they're outcompeting the water. pH, this is another way. The excess OH or pH ions are going to react with those side chains. Remember the zero ion, it's positive and a negative. That's a lot of the interaction that's happening with that. If you start messing with that by messing up the pH, all of a sudden you're going to have a bunch of negatives or a bunch of positive or a bunch of hydrogen ions running around. It's going to mess with everything, and that is going to mess with primarily the salt bridges. Salt bridges. That's primarily what's going to mess with that. Those salt bridges that are coming up. Inorganic salts. So a high concentration of salt is again going to mess with high concentration of ions. Can disrupt the salt bridges. So the salt bridges are really important. We talked about that in secondary and tertiary structure. Denatured proteins lose biological activity and are usually less soluble. soluble. Okay, we're gonna have to make another video. I'll see you in a minute.